Okay, so we are in video lecture 2 of 3 for our cell cycle and mitosis videos. So at this point, before you start this one, you should already know about the different forms that DNA can take because we're going to start keeping track of that DNA, talking about the cell cycle here. So in this video, we're going to think about starting with the question, why do cells divide? Like, why are we talking about this to start with? And then we'll get into the big picture view of an overview of the cell cycle. So this first question, why do cells divide? When we think about organisms growing, that's one obvious place where we would need new cells, right? So if this is a baby goat is going to turn into an adult goat. Adult goats are bigger than baby goats, and they need more cells to make up the organism. As organisms grow, they add on new cells rather than having their existing cells get really, really huge. We talked about how cells need to have not too much volume with a lot of surface area so that they can exchange nutrients and waste with their environment. And because of that reason, as organisms get bigger, they add on new cells. So any organism that's growing physically or developing is going to need to make new cells. We also have certain tissues where we need to replace a lot of cells. So the turnover rate might be really high when cells become damaged or if they die. So, um, and some examples of this in our bodies are that our skin cells are often just sort of the word everyone uses is sloughing off. And as they do so, we need to make new skin cells, right? And the same thing is true for cells that line, for example, our intestinal tract. And this is a cell that would line your intestine. These cells are there's stuff brushing against them constantly, and because of that, their turnover rate is quite high. So when we're replacing cells, that's another reason when cells will need to divide. And then last but not least, we have asexual reproduction. So this is, I think, most obvious in the case of single-celled organisms, where some bacteria, they're just going to divide by splitting themselves into two. But it also is the case for some organisms that are a little bit more complex. So this is a picture of what's called a hydra, and you can see that it's got a little foot here and it's got some head region up here. But what's happening right here is a new hydra is budding off, and this piece is going to become a brand new hydra. And so this organism is one that at least has the capability of reproducing asexually. So this is happening all through just simple cell division. And so in each of these cases, our goal is to produce daughter cells, which is the daughter cells are what we call the offspring of a cell. If a cell splits itself into two, the two cells that result are called daughter cells. And these daughter cells are genetically identical to the parent cell. That's what we want. So if we're making more intestinal cells or if we're making more goat cells, we want cells that are genetically exactly the same as the cell we started with, right? We want them to have the exact same genetic information. So it's really important that each of our daughter cells is going to get all of the chromosomes. No chromosomes are missing, but also no extra chromosomes. We want exactly the right amount of chromosomes for these daughter cells. Okay, so that's our sort of why, and now let's start to look at how. How do cells divide, and what's the process um, before they divide? Like, what's their life cycle look like in general? So this is a diagram showing what's called the cell cycle, and what the cell cycle is really just the life cycle of a single cell. And typically, we tend to think of it as starting when a cell is formed and ending when the cell divides. And it might seem a little bit arbitrary to say that it's a cycle, but it has a start and an end. But if you think about it, if a cell starts here and then ends up dividing, now each of the daughter cells is going to go through the cell cycle anew, right? So it still is a cycle, but it's just going to restart again every time daughter cells are formed. And that sort of is the end for our original cell once it divides and turns into two new cells. So when we're thinking about a cell's life cycle, there are two big distinctions that I want to, or not distinctions, two main phases that I want to draw your attention to. So, one phase is called interphase. An interphase encompasses every part of a cell's life cycle where it isn't actively dividing. So when a cell is just growing, when it's doing its thing, that's all called interphase. An interphase has three main parts that make it up. Two of these parts are called G1 and G2. And the G1 and G2 phase is the cell is just kind of it's growing, the cell is living its best life. It's just doing its own thing. So the cell is maybe making proteins. It's carrying out whatever function it needs to carry out. So if we're thinking about a skin cell, it's acting as a barrier. If it's an intestinal cell, it's taking up nutrients from the food that the organism has consumed. So it's just doing its cell stuff in G1 and G2. And then in the S phase, this is really important. It's when the DNA is replicated. So we talked earlier in that first video that DNA can be either replicated or unreplicated. And this S phase in interphase is when the DNA is copied. So now maybe looking at this diagram, it's all becoming maybe a little bit more clear that this cell cycle is going to have a section in it where the cell is going to divide and turn into two new cells. So in order to do that, we need to double our genetic information, and that happens in the S phase. And then, so interphase, we've got G1 followed by S, where the DNA is copied, and then followed by G2. And then the other section, the other main part, is the M phase, which stands for mitotic. So this is when the actual chromosomes are divided up between daughter cells, so the actual division of our nuclear material. So we copied our DNA in the S phase, but the actual division of the chromosomes between those two daughter cells is going to happen in the mitotic phase. And then lastly, one little quick note, it's a little bit covered up by these video controls, but some cells don't actually, their dividing is not a big part of what they do. So a lot of neurons in your brain, a lot of retinal cells, a lot of cells in your inner ear, these are cells that once they're formed, they don't really die, but they also don't divide. So some cells are going to just sort of stick around in the G1 phase if they're not actively dividing cells, and they're just sort of destined to be a non-dividing cell, kind of stick around right here. Okay, so let's think about these different stages of the cell cycle in terms of what the DNA is doing in each of these stages. So here's a picture of how we can think about the DNA at these different points in the cell cycle. So first here in G1 is kind of be our sort of starting place. And you can see here that we've got 5 molecules of DNA. 1, 2, 3, oh no, 4, I can't count. 4 molecules of DNA, 1, 2, 3, 4. But none of the molecules are replicated. They're all just single copies. So they're originals, they're just one piece for each one. And then as we get to the S phase, this is where we're going to replicate our DNA. And so you can see that now all of the DNA has two copies, or each of the DNA molecules consists of 2 copies. So we still have 4 chromosomes. We don't count each sister chromatid as a separate chromosome. We still just have 4, but each of the chromosomes we now would say is replicated. So we have 4 replicated chromosomes. And then finally, as we get into the beginning of mitosis, the DNA is going to go from that chromatin form where it's sort of unwound and loose, and we said it was like spaghetti, and it's going to condense into our chromosome form, which is our tightly packed DNA. So it's still going to be in its sister chromatid form. We're still replicated, and then eventually, as we go through mitosis, as we start to divide up the DNA into 2 separate daughter cells, we're going to pull apart these sister chromatids and sort of divvy them out between the 2 daughter cells, so that each daughter cell ends up with exactly the same amount of DNA that our parent cell started with. Okay, so we've talked about G1, S, and G2, and I want to just give a super, super big picture overview of the stages of mitosis. And then in our last and final video, we will go into much more detail about the particular stages of mitosis. So one note before I name these phases, I want to say that mitosis is all about the division of the genetic material. So when we're thinking about the mitosis and the stages, we're really paying attention to what the chromosomes are doing, and how they're being organized and how they're being portioned out to both of our daughter cells. Obviously, in order for this to happen, there's things that are happening with the cytoplasm, with the cell membrane, but what we're really paying attention to is what's happening with the DNA. Okay, So I cut off the beginning of this diagram, which started with interphase, because we already sort of talked about that. So we're starting just with, this is all part of the M phase, of the mitotic phase. So our first stage is called prophase, and we can see our chromosomes here. And then I'll go into much more detail about these in our last and final video, so I'm just kind of naming them here. So we start with prophase, and then you can see that in metaphase, our chromosomes are moving to the middle of the cell. And then in anaphase, hopefully you can see the big thing that's happening here is that our sister chromatids are getting pulled apart. So now we just have individual chromosomes at this point. And then our last phase of mitosis is called telophase. And that's where we're reforming the genetic material but now it's in the form of two separate daughter cells. So two individual nuclei. Okay, so like I said, I know that this description was super, super broad, and you'll have to watch the next video for our explanations for what's going on in each of these stages.